All right, chapter 4, let's go ahead and dig right on in. So with chapter 4, we're going to deal with DNA and RNA and the importance of both. We know that so many CSI shows and Law and Order and criminal shows have been created because of DNA. People have been exonerated because of it, and people have also been convicted of it. So the first thing we need to know is that DNA stands for deoxyribonucleic acid. We have 46 chromosomes. 23 come from mom, 23 come from dad. And with this DNA, we know that its structure is going to consist of a sugar, a phosphate group, and a nitrogenous base. So what we have is 5 nitrogenous bases on this page, but DNA only subscribes to 4. So we have adenine and guanine, which is abbreviated adenine with an A, guanine with a G, cytosine with a C, thymine with a T, and uracil with a U. Now the way to remember which base pairs belong to, which base pairs belong to which subgroup, purines, the way I like to remember is purines are pure as gold. So purines, P-U-R, as gold, and pyrimidines are cut, C-U-T, cut the pie. Now on this page it says purines have a double ring and pyrimidines have a single ring. So if we look at the structure here, we see that adenine, which is a purine, has 2 rings. 2 rings here, 2 rings here, whereas our pyrimidines only have the 1 ring. So DNA has some unique characteristics. The structure of DNA is in a double helix. It looks like a spiral staircase. This is how they pair together. A pairs with T and C pairs with G. This is the law of complementary base pairing. If this is, for some reason, A pairs with G, then we know a mutation has occurred. So anytime A and T combine together, there are 2 hydrogen bonds. And if C and G combine together, there are 3 hydrogen bonds. So here we can see A and T, there are 2 hydrogen bonds, whereas we can see G and C, there's 1, 2, 3 hydrogen bonds. And then we see the spiral staircase. Now, a gene is a segment of DNA coding for synthesis of a specific protein. The whole purpose of the Human Genome Project is to unlock the secrets to every gene, to see where we can find cancer, to see where we can find Parkinson's disease and Alzheimer's and sickle cell, with the thought of being able to cure these diseases, which of course would cause all of us to live a lot longer and fuller lives. DNA was discovered by several scientists. A Nobel Peace Prize was given to Watson and Crick and Wilkins. Now, Rosalind Franklin was a woman, and she was the one that actually discovered the double helix. She died of radiation, but they never gave her credit for her work. They took her work and presented it as their own. We're going to skip through some of this. I do want to bring to your attention what we did in lab. Remember, the chromatids were the single pieces, the sister chromatids on the side and the centromere in the middle. So all this is saying is how we started with this large helical structure and how we broke it all the way down to what you can see under a microscope. Now, again, with DNA, most DNA is found in the nucleus, and every time it's about to divide, the cell is going to make a copy of them. So here we have the sister chromatids, what we looked at, the centromere, which was that metal piece that you put in the middle. Now, going on to RNA, RNA structure is similar in some ways to DNA and different in a lot of ways as well. It does not have that double helix, so it is single-stranded. And it is missing the thymine base pair. It replaces it with uracil. Now, RNA is found mainly in the cytoplasm. 3 important RNAs for this subject matter for RNA is going to be messenger RNA, ribosomal RNA, and transfer RNA. Now, the base triplet, there's a sequence of 3 DNA nucleotides. When they are together, they are called the codon. The codon is what you see in messenger RNA. There are 64 possible codons available that represent the 20 amino acids. But I only require you to know 3, which are the stop codons and 1 start codon. So there's only 1 start codon. The way I remember that is all school starts in August. So the start codon is August. Stop codons, UAG, UGA, and UAA. I like to think of them as schools in Georgia: University of Georgia, University of Albany, Georgia, University of Atlanta, Atlanta. Of course, these 2 I have made up, but these are how I remember what's going on for the stop codon. So at the end of a message, it's going to cut it off and say, no, we do not need this. Now, all body cells, except sex cells and some immune cells, have identical genes. Identical. So when a gene is activated, messenger RNA is made. So the process of protein synthesis goes like this: from DNA to mRNA to protein. Messenger RNA is called messenger RNA because it carries a code. That code is going to code for a protein. It's going to give the recipe on how to make the protein. So to go from DNA to mRNA is a process called transcription, and this occurs in the nucleus. To go from mRNA to protein is called translation, and that occurs in the cytoplasm. So make sure that you know those 2. Transcription is DNA to RNA. Translation is mRNA into a protein. All right. So transcription just copies the genetic instructions from DNA to mRNA. So DNA is the blueprint, and mRNA is a copy of the blueprint. The mRNA then takes the copy of the blueprint and makes the new building, and that is the protein. So there's RNA polymerase that is present, and what it does, it binds the DNA and helps to assemble mRNA. So it kind of calls everything into order so that everything can start. This we already know. C goes to G, G goes to C, T goes to A. This is the key. But when A is on the DNA scale and we're trying to convert it, A goes to U. So if we have, for example, TA, 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 and we're taking this and we want to make a copy of mRNA, so we're going to say T goes to A goes to U, T goes to A goes to U, T goes to A, and A goes to U. Okay, so let me see if I can put that in so you can see that. So if we were going to mRNA, that would be A, U, A, U, A, U. We're going to skip past this and continue on. Next part of translation, which is very, very important. Remember, translation is going from mRNA to protein. So the mRNA is going to go from the nucleus to cytoplasm because remember, everything started in the nucleus. Transfer RNA is going to take that coded message and bring it to a single amino acid to be added to a peptide chain. So it's like a little choo-choo train. It's transferring from one area to the protein chain. Once it gets to the protein chain, it's going to give it to the ribosomes. What the ribosomes do is they read the message. And we talked about ribosomes before as one of the organelles, and it's found they float freely in a cytosol. You also see them on a rough ER. You see them on a nuclear envelope. You see them in several different areas. So again, with mRNA, we start with AUG. School starts in August. That's how we're going to start the process. Transfer RNA is going to pick up the free amino acid and stick it to the protein peptide chain. Okay, so mRNA takes it. So we can go back. Went the wrong way. All right, so what we're going to do, the mRNA is going to take it, and we're going to hit protein synthesis. It's going to take it from the nucleus, bring it to the cytoplasm. Transfer RNA is going to take that amino acid and take it to the cytosol. Okay, the amino acid is going to the cytosol because in the beginning we started with the nucleus. Okay, so here's our overview. We have the DNA double helix. We break it apart. And now we're going to start transcribing and translating. So to go from the DNA strand to the RNA strand, that is translation. So T goes to A goes to U, C goes to G, C goes to G, G goes to C, C goes to G, and so on and so forth. We see right here we have the start codon because school starts in August. We also see that we have a stop codon here. Here's where the transfer RNA comes in. The transfer RNA is actually an anticodon. This strand right here is called the codon. So to go against that, you just translate another way. So we go A goes to U, U goes to A, G goes to C. So the key piece that I want you to know is at the bottom, these little abbreviations are amino acids. So the transfer RNA is taking these amino acids and is going to add it to this chain. So the amino acids carried by these 6 TNR molecules is going to add it to this chain. Once it adds it to the chain, then the ribosome can read it. The ribosome can read it and we can get the protein that we need because the end result is to have a new protein. Okay, so along the way, we have to have some type of monitoring system to make sure that everything works. So we have some chaperone proteins, and they pick up new proteins and make sure that all the proteins are folding the way they are supposed to. If not, we get rid of them. Proteins are to be used in a cytosol are likely to be made on free ribosomes because remember we have free ribosomes in a cytosol kind of floating around. Then we have other proteins that go to the Golgi apparatus, which we talked about, because they are the UPS of the cell. They ship it, they sort it, they package it. So we have 2 different destinations. When we're looking at the ER, the endoplasmic reticulum, especially the rough ER, is going to, again, remove some of the amino acid segments, is going to add some pieces to it to make sure that it is properly situated so that it can be the protein that the body needs. Gene regulation. Genes can be turned on and off. We know that about cancer genes. We call them oncogenes, and how we can turn them on and off. Okay. Liver cells turn off hemoglobin genes, so some work with each other. And these are usually controlled by hormones. Example, mammary gland cells turn on a gene casein protein only when breast milk is produced. Now the fun part that we've done in class, DNA replication and the cell cycle. So before it divides, it has to duplicate. So DNA unwinds from histones, and that is an enzyme called helicase. And I need you to know that helicase. Helicase unzips the DNA because remember, it is a double helix and we need to break it apart. Then we have that polymerase that comes in is going to make sure that it is right to transfer to mRNA. Then we end up with new DNA repackaged. We're going to skip through this part here, but this is what we start off just to give you an example. Parental DNA, it is, you can see, wound. We put DNA helicase through it so that it separates and they go through 2 different strands. We have an old strand and a new strand, and when that occurs, we call that daughter DNA. Then we glue it back together with something called DNA ligase. DNA polymerase does make mistakes, so there are tons of errors in replication areas, and a lot of times the body does catch it, but there are some areas that it does not, and when it does not catch it, that's when we end up with mutations. Some mutations are extremely harmful. They cause diseases, they cause malfunctions, and some are very silent, nothing changes. So looking at the cell cycle, we go with interphase, prophase, metaphase, anaphase, and telophase. We talked about that. Another acronym to remember the order is I pass my anatomy test. We talked about G1, S, and G2 and how both of these are growth areas, and the S stands for synthesis. So here we go. Growth and normal metabolic roles, DNA replication, growth and preparation for mitosis. So we see that interphase is the longest phase. If you think about I pass my anatomy test, I is the longest part because in order to pass your anatomy test, spend a lot of time studying. Then we go to prophase, metaphase, anaphase, telophase, and cytokinesis. What we already said, this is just going to be growth. The most important part I need you to know for the synthesis phase is DNA is going to replicate. G2 is going to continue to grow and prepare for mitosis. The 4 phases, we talked about this. The prophase is where you see that the nuclear envelope has now disappeared. There's no plastic bag. See, nuclear envelope disintegrates. Metaphase, the chromosomes are aligned on the cell equator. They meet in the middle. So prophase, over here. Metaphase, the chromosomes come towards the middle. Anaphase, the chromatids, they are apart. Okay, they become antisocial. They're two different ends. And then telophase, at the very end, we see that the mitotic spindle disintegrates, chromosomes cluster on each side of the cell, and a new nuclear envelope begins. Okay, chromosomes uncoil to chromatin. Okay, so they're no longer joined together. They're completely separate. And you saw this when we were doing it in the lab. Cytokinesis, I do need you to know this word. Cytokinesis is the division of the cytoplasm into 2 cells. So it pinches off and it creates something called a cleavage furrow around the equator of the cell. If I can show you. Here we go. Cytokinesis, we see that pinch right here. Regulation of cell division. Cells divide when they have replicated their own DNA. They have enough nutrients, chemicals, signals. When there is space, when there is no space, we are not going to divide. Cells stop dividing when they're too snug, there's no room, or if there's no growth factors. Now, last but not least, the fun parts, how we can look at chromosomes and heredity, how we are made up. So we're going to start with the karyotype. A karyotype is simply a chart that lays out all 46 chromosomes. You have 23 pairs, 2 members of each pair. So 22 pairs are called autosomes, which means they are not sex genes. And you have 1 pair of sex genes. Female has an X chromosome. Together we know that's XX. Male has X and a Y. So here is a karyotype. So we can look here. So if someone has Down syndrome, or we say trisomy 21 on a karyotype, you would see another chromosome placed right here. And we know that this is just an example, but if this was crossed out, you could just look and see if there's 2 X's or if there's an X and a Y to determine if the karyotype was a male or female. So diploid. Diploid is any cell with 23 pairs of chromosomes. Haploid are 23 unpaired. Okay, so 2N, so that would be 23 times 2. Haploid would be 46. Fertilization restores diploid numbers, 2N. Okay, so before haploid just 46, diploid 23. So right now we all have 43 chromosomes. When we mate with another partner, reproduction, that number gets half. Because if we were to give our child 46 and your partner gave 46, that's way too much DNA for one person to have. Because one person is only supposed to have 46. They're supposed to take 23 from mom and 23 from dad. Genes and alleles quick run through with these. Anytime you see a dominant allele, you know it's going to be dominant because it has a capital letter. As a capital letter is going to hide the recessive allele. Recessive allele always represented by lowercase letters. Genotype and phenotype are another part that are added. Genotype is what the gene actually looks like. Phenotype is what I can see. If you have brown hair, if you have curly hair, if you have brown eyes, if you're tall, if you're short. So a phenotype tells me physically what you look like, whereas a genotype tells me what genes are involved. And we use a Punnett square to figure these things out. So here we go. Homozygous, heterozygous. So homozygous is when both letters are exactly the same. Either we have two capital letters or we have two lowercase letters. So in this one with the cleft chin, this is homozygous because we have two capital letters. Two capital letters are also going to tell us this is a dominant allele. So capital letter. And then we have here a heterozygous because we have a mixture. We have a capital letter and we have a lowercase letter. This is heterozygous. And because there's a capital letter, it overpowers that little c. So it's almost like it hides it and you don't see it at all. So when we mix this together, homozygous cleft chin, heterozygous cleft chin, heterozygous cleft chin, and homozygous. Homozygous because they're two little c's. Everyone see that? So with this Punnett square, what we do is we just simply multiply each one. And we'll show you on the next one. Parents can be healthy, heterozygous carriers of hereditary diseases. So we have genetic counselors that will test the amniotic fluid of the baby to see if a mom is at risk for a baby that, let's say she is of mature age, and according to all statements, of medical statements, a woman 36 or over is considered matured or advanced prenatal history, prenatal candidate. Okay. So multiple alleles, codominance, and incomplete dominance. I have a better PowerPoint that I'm going to switch on so you can see that one too. So we'll go through that, and it has more that you can click through. Let's see. Polygenic inheritance just means there's more than one gene that contributes to eye color. If you think about it, we are all fascinated by mutations in the body, which is why we have all these superhero movies like X-Men. We have Teenage Mutant Ninja Turtles. We have Iron Man. The whole Marvel series all came Spider-Man because of some kind of genetic mutation. There are some sex-linked traits, which means it only occurs in certain groups of people, like color blindness. Usually only occurs in men. Hemophilia almost always occurs in men. So let's look a little closer here. So we have XX, which is female, and XY. The X's are the ones that carry all the information. So let's say that this X is bad. If that X, because mom is going to give an X to her daughter or son, either way. Mom gives that X that's bad over here. There's not another X to mask that. So this will show up. Whereas over here, we have another one that's good. I think they reversed it in this one. They made this one the bad one. So this is the good one, and this is the bad one. So the good one can overpower the bad one, where over on the male side, that's not the case. We only have one copy of the X, and the Y doesn't do much to help this out. So that child ends up being colorblind. Population levels, we'll skip through that. I want to skip through. Oh, I do want to talk briefly about this. Cancer. Malignant tumors can cause cancer. And what it says is it metastasizes, which means it moves throughout the body. It does not stay in one area. So it moves throughout. Oncology is a big specialty when we start talking about cancer and how sometimes DNA is not able to check itself. When it's not able to check itself, then this is what happens. We have DNA that continues to replicate and goes rogue. Let's see. Only 5 to 10% of cancers are hereditary. You think about things like breast cancer or prostate cancer is usually hereditary. But then there are some carcinogens that we get from grilling on the grill with burned hot dogs, or x-rays, or chemicals like cigarette tar and food preservatives that are cancer-causing agents that have nothing to do with your inheritance from a parent or a family member. But carcinogens can trigger these gene mutations. So we have oncogens, which I've talked about before. They can accelerate out of control. And gratefully, we do have some tumor suppressor genes. When activated, they can help silence the oncogens, but not all the time does that work. All right, so I'm going to switch so you can see this other one that I have. All right, I'm going to upload this genetics PowerPoint that I think is a lot easier to understand, to just kind of add, to go through. I really encourage you to go through this one. It is so self-explanatory. It goes through dominant and recessive genes, and it even has problems that you can go through. Okay, so it lets you figure out which one is which and how to go through what double and recessive genes look like. Okay, so dominant and recessive. If you have any questions, please let me know, and I will see you next Wednesday so we can finish this up. Okay, have a great day.